Right, hello, welcome to the second video. We have moved the brain to the yellow one, so that makes you know that things are getting a little bit more complicated. So, concepts of choreoathetosis and ballismus originating in the basal nuclei. So before we go into the neuroanatomy of why it happens, let's quickly define these four terms. So when we think about movement disorders, we can classify them based on how kinetic they are, aka how much you move. Is it hyperkinetic or hypokinetic? And a few different things here to be aware of. Chorea, sudden fast distal movements, versus athetosis, slow writhing distal movements. Okay, so choreoathetosis is often intermingled, such as in Huntington's disease. A dystonia is a slow, long-term movement involving an entire muscle group. And that can be like a dystonic contraction. Carpal tunnel is an example of that. Your ballismus, on the other hand, is not distal like choreoathetosis, but it's proximal. So the top of the upper limb, the shoulder, will fling out if it's a hemiballismus, and the hand will just suddenly abruptly zip out like that. That's your fast proximal limb movement. So looking at the neuroanatomy of these hyperkinetic states, choreoathetosis is due to this. So our putamen is decayed. Okay, so because of this, we lose the indirect pathways regulation. Remember, the putamen is integral to the indirect pathway. Review that from the previous lecture. So because we lose the putamen, ultimately we lose the handbrake of the subthalamic nucleus. Okay, so there's excessive inhibition of the subthalamic nucleus, which means that there is an excess excitation in the thalamocortical pathway, which means that movement that should be vetoed by the basal ganglia, the basal nuclei, is not vetoed. Hence choreoathetosis. Now, ballismus, a little bit different. That's a specific lesion to the subthalamic nucleus. So it has a similar outcome, but that's usually a vascular cause as opposed to a neurodegenerative cause. And that again causes excess in the thalamocortical pathway, so things are not being vetoed, and that's when you get the proximal flinging. So it's very important that you can define choreoathetosis, ballismus, and dystonia, and that you know the neuroanatomy relative to choreoathetosis and ballismus. Loyola University did a very good neurology primer, so if you Google Loyola University Disorders of Basal Ganglia by Dr. Merschet here, there's a few little pages there if you want to know a little bit more about it. Now, the septum pellucidum. So, where is the septum pellucidum? Well, the easiest way, remember the corpus callosum? Here's the corpus callosum, here's the corpus callosum. Just inferior, that's where it lies just inferior. So the septum pellucidum is this thin, transparent membrane in the midline, right here. Septum pellucidum, or septum lucidum as it's called there. So we have the fornix underneath, we have the corpus callosum above. Okay, here's the choroid plexus, which is quite interesting. So here's the fornix, and then you've got the fimbriae here, and you'll have the amygdala down here. You've got the hippocampus as well around here, but the septum pellucidum is between the corpus callosum and the fornix. Now, worth noting is that between the corpus callosum and the fornix, and it's cavitated in babies. So you might see on an axial CT or MR of a younger person, a baby, or it's very variable as to when it closes, when the white fibre tracts form around it and things, but the septum pellucidum is a hole that you will see between the lateral ventricles on either side. So that's septum pellucidum. So number 3 is the insula. And now we touched on this last time. If you take the lateral sylvian fissure and open it up, you've got your frontal operculum, your parietal operculum, Heschel's gyrus, which is the transverse temporal gyrus, which is the primary auditory cortex. And within here we have the insula. So peel open that lateral fissure and you come to the insula. That's where it is. If we do a slice here, beautiful little cross section, we've got our caudate nucleus, we've got our putamen and our lentiform complex, and there's our thalamus. Move aside here, we've got the internal capsule. We'll have the external capsule and other things here that we'll come to later. But here is the insula. So here's your lateral fissure, peel this backwards, then you'll find your insula here. So that is the anatomy of the insula. And here's another cross-section with a little bit more detail. And I want you to note, in fact, some things here. Oh, no, that's a bit later on, don't worry about that. But again, this is a structure, this is a cut that you should be very, very familiar with. And I mean really familiar with. So spend some time, cover up these labels and make sure that you cannot just really quickly zip through all these different structures. And the same with this cross-section here. So we've got the caudate, we've got the corpus callosum like this, we've got the third ventricle here, and we have lateral ventricles either side. Here's our fornix, here's our corpus callosum, which means that the connecting between the two, we've already done that, that's our septum pellucidum, remember? So we have our thalamus, internal capsule, lentiform, this this pinky area here, that's the claustrum. And so medial to that is our external capsule and then our extreme capsule. So we have three capsules, internal, external, claustrum, extreme, and then immediately lateral to that is the insula. So it's worth being able to name all of these different things, sort of like lateral to medial. Insula, extreme, claustrum, external, the lentiform complex, the internal capsule, the thalamus, the third ventricle, and then it repeats. So, the insula can be divided many ways, but one of the most common ways that it's divided is the anterior, middle, and inferior insular cortical regions. The anterior one is mostly processing information that is integrated from the different lobes by the posterior and the middle. So you tend to get afferent input into the posterior and into the middle, and then decisions are made in the anterior, which is then fed out. Von Economo neurons are a very important thing that have been found. They're pretty much exclusively found in this region, and they're pretty exclusive to higher order primates. There's a few in elephants and some dolphins as well. They seem to be very correlated with intellect. They're also, interestingly enough, targets for frontotemporal dementia. And so you will get wastage within this region quite early on in that particular nasty type of dementia. So, a quick review from 2017, which is worth a read, but I pulled the most important quote to do with the function of the insula to fill our learning outcome, is that the anterior insula is a core in supporting subjective feeling states. So it regulates the introduction of feelings into cognitive and motivational processes. So it's a sort of like injection, it's a syringe of feeling into a cerebral act or a motivational process. Okay, that's the role. It's also widely thought of to be something to do with shame. It's bringing about that motivational correlate and the shame that triggers a motivation of either running away or doing something. Shame and fear. It's plugging all of these specific feelings into your day-to-day life. So, lesions to the frontal eye field, dominant angular gyrus, and these association areas that we talk about. So, if I can make that spin, Brodmann's area number 8 is the frontal eye field. Oops, there's a frontal eye field. Now, ordinarily, this frontal eye field stimulates the contralateral parapontine reticular formation to cause the eyes to look the other way. Revise your brainstem and conjugate deviation notes from block 3 for that, which we might go into a bit more detail in the brainstem. The consequence of a lesion to your frontal eye field is a unilateral deviation toward the lesion. So your eye will move toward the lesion. Now, interestingly enough, there are reasons no one truly understands. There are theories, but they're not that satisfying. Is that if you stimulate the frontal eye field in neurosurgery, you get deviation to the opposite side. So, a lesion, deviate to the ipsilateral side, stimulation to the contralateral side. And that's where it is, basically. So, this is worth knowing, just reminding yourself of Broca's and Wernicke's area. So Broca's, which is 45 and 44 here. Follow the lateral fissure, and you find the supramarginal gyrus, and then you find the angular gyrus. The posterior portion of 22, and then the transverse temporal or Heschel's gyrus. This is the complex of Wernicke's, this receptive component of speech and prosody. This is the expressive portion. Okay, so make sure that these are something that you're very, very familiar with. Now, the MCA, the middle cerebral artery, comes out of the lateral fissure about here and gives a superior branch and an inferior branch. And the superior MCA goes to Broca's area. And the inferior MCA goes to Wernicke's. Now, the more proximal portion within that fissure will supply the arcuate fasciculus that connects Wernicke's and Broca's together. So MCA lesions are going to be, MCA infarcts and things, proximal to the fissure when the superior and inferior divide happens. So proximal to that, going to affect the arcuate fasciculus and receptive and expressives, causing those named aphasias. But if you have a distal infarct to the superior MCA, then Broca's will be degraded, the arcuate fasciculus will not, and Wernicke's will not. So if you have a Broca's without a Wernicke's in a stroke patient, then you can assume it's in a more distal aspect of the superior MCA branch on the dominant hemisphere. So, the angular gyrus, if you follow the superior temporal sulcus posteriorly, then you find this angular gyrus here. Now, the angular gyrus in the dominant region has this thing called Gerstmann syndrome. Gerstmann syndrome is a triumvirate of things, which is issues with arithmetic, issues in devising left from right, and issues in your finger recognition. This is known to dramatically underfire in dyslexic persons, so make of that what you will. But that is the consequence of an angular gyrus lesion, Gerstmann syndrome. Nice and high yield, that. Now, these association areas that we're going to talk through right now are ones that are specifically mentioned. So the dominant 5, so BA5 and BA7. So I want you to think about the dominant region of BA5, the superior parietal lobule up here, ideomotor apraxia, and astereognosis, right? And astereognosis in particular is very easy to test. You put something in a patient's hand and you tell them to tell you what it is, so a watch or a pen or something, and they close their eyes, and if they're not able to do that, then they can't sort of like judge this stereotactic sensation. Now, the non-dominant hemisphere, the other side, is involved in depth perception and relation in space, so you will get neglect syndromes, hemispatial neglect, where the contralateral visual field will be completely ignored and will be neglected, and they won't even be aware of the neglect. So, V2 and A2, these auditory lesions, and you can imagine what the deficiencies are, right? So it's a neglect syndrome as well, in which they can hear it, but they're not sort of attuning a sense of importance to it. So it's there, and they're receiving it, and the auditory centers, the primary visual centers light up, but the actual response isn't a thing. Now, this is very theoretical, because it's very unlikely that you'll get a V2 lesion without a V1 lesion or a V3 lesion, just because it's all in such close proximity. And if you did see that, it would likely be written up in a journal as a once-in-a-career thing, really. So this is a summary of the different cortices that we've gone through. And I'm not going to go through them again. This is wrong. Wernicke's, we know, is a bit more extensive. Broca's is a bit more extensive, as we've been through. The primary auditory cortex isn't that far out. It's more inside the lateral sylvian fissure. Also, you need to be aware that the secondary somatosensory area is basically this posterior parietal lobe. Okay? So, the claustrum, which we briefly touched upon already. Here's the claustrum, that region here. So it runs between the extreme and the external capsule, lateral to the putamen. Okay, so that's the claustrum. So putting these pins in here, a little spotter for you, you should be able to identify each of these structures. So if we go, let's say this is number 1, let's say this is number 2, So this is number 3, this is number 4, this is number 5. Pause the video now and try to work out which is which. So 1 is your insula, 2 is your extreme capsule, 3 is your claustrum, 4 is your external capsule, and 5 is your internal capsule. So just as a recap, some active recall, can you pause the video now and tell me the blood supply and the contents of all the different portions of the internal capsule? And can you tell me how the putamen and the globus pallidus, externus and internus function differs in the direct and indirect pathways? So, the association fibre bundle of grey matter interconnecting frontal, parietal and occipital lobes. What is the function of the claustrum? So, no one really knows, but it seems to be a very interconnecting component of various different lobes. Basically, almost all of the brain talks to the claustrum. And that led Crick, or Francis Crick of DNA, you know, nicking Rosalind Franklin's research on photography. Not a big fan of Crick, but he postulated that it was a possible origin for integrating conscious perception. So it may be where this whole idea of the major locus of consciousness is, or it's certainly a big component in the orchestration of consciousness. Not sure what we think about that nowadays, but no one really knows what the claustrum is, but it often comes up in neuroanatomy because it's such a well-defined structure. Now, we're not going to go into all the different functions of these cells histologically, but what I do want to lend your focus to is that the neocortex, this outer shell of the cerebral cortex, is split into 6 layers. You've got your molecular layer, and then you have 2 granular layers and 2 pyramidal layers, internal and external for each. And you have a multiform layer here. It's called multiform, down here, the deepest layer, and this is the most superficial. It's called multiform because it has loads of different cells, granular, fusiform, stellate, loads of cells. Don't worry about knowing them. Seriously, don't. Layer 5, this pyramidal layer, is full of these pyramidal cells. Remember, we've talked about these before. And they send out their dendrites laterally like this. And the consequence is they have a banding here. And the granule cells do a similar thing, and there's a second band. And these 2 bands are very well known. The outer Baillarger and the inner Baillarger layer. So remember, here's the outer portion, here's the inner portion going deeper. So here's your inner band and here's your outer band. And that's a sort of way to nicely differentiate the few layers that we see. The external pyramidal layer has a few more pyramidal cells, but not extending wide enough to give us a 3rd band. The external granular layer the same. So, Betz cells, which are giant pyramidal cells, are mostly seen in the internal pyramidal layer, and stellate cells, which we'll come across when we do the cerebellum, are seen in this internal granular layer. You don't need to take much from this. I doubt there will be many histology questions, and functionally it's not that important unless you're going to do a PhD in the neocortex. But this is just something to be aware of. There are 6 layers. There are 2 horizontal bands for these longitudinal columns. 2 of the big cell structures you need to know are pyramidal cells and granular cells, Betz cells and stellate cells. Then there's a mixture of these cells, apart from the pyramidal cells, down in layer 6. This is the reason I mentioned you don't need to worry too much about it, because although I've mentioned 2 or 3, and although there's 4 or 5 in the learning outcomes, there are so many different types, and there's a lot of variation. So there's Martinotti cells, they're usually found in layer 1 up here, but they can be down in layer 5. There are chandelier cells that hang over in layer 1, 2 and 3 and then hang down here. There are bipolar cells, there are tuft cells, multipolar cells all over the place, and then basket cells, small and large and nest and multiform, and all manner of different cell types all over the place. The 6 layers do tend to persist in most areas. There are a few notable exceptions, like the primary motor cortex that drops a layer, but functionally, is it that important to us? Not right now. Here's a Betz cell. This is a pyramidal cell. So, little pyramid here. It has loads of dendrites, and from its base, here's the axon. So we have all of the information coming in from different dendrites and summating, temporospatial summation. Here's our pyramid, and then the axon is a singular baseline. Okay, and there's the axon. That's what it actually looks like. This is a Martinotti cell, or a chandelier cell, one or the other. So here's your top layer, and it's descending down from that molecular layer down through all of those granular and pyramidal external and internal layers. And that's that. So that is all the learning outcomes addressed. So, back to the top. Yes, do not worry too much about this histology. It isn't that useful, and the cortical potentials are more neurophysiology, so I wouldn't worry too much about that either. But that is the basically all you need to know for the cerebral cortex. So next we're going into a deep dive of the subcortex.